what other kinds of alterations can we have? Inversions. An inversion simply means that part of a chromosome has been cut out, turned upside down, and then stuck back into the chromosome again. In this picture, the part of the chromosome between the dashed lines has been cut out, turned upside down, and stuck back in again in reverse order. This is called an inversion. Now, such inversions themselves usually don't cause problems because all the genes are still there. They're just in the correct order. Unless one of these breaks occurs in the middle of some important gene, this person is likely to still be okay. However, sometimes such an inversion can be lost. If that inverted part is lost, then that produces a deletion, and the deletion then can cause problems in the patient. So these are inversions. This particular example is known as a pericentric inversion because the part of the chromosome that is inverted includes the centromere. It would be called a paracentric inversion, P-A-R-A, paracentric inversion, if the centromere were not included in the part that is inverted. So we can have pericentric and paracentric inversions. But this is not associated with a lot of famous diseases that I know about, so it's somewhat less likely to be on your exam. We can also have ring chromosomes. A ring chromosome means that the two ends of a single chromosome have joined together to form a circle. This particular picture shows a ring X chromosome. Now, again, the ring chromosome itself is not likely to cause significant problems, but one of the things that sometimes happens is that sometimes a ring X chromosome can be lost from a cell. So just suppose we had a fertilized egg that had such a ring X chromosome, and then the ring X chromosome was lost from that fertilized egg. That could then result in Turner syndrome. So this is an alternative way of producing Turner syndrome. The last kind of structural alteration illustrated in this chapter is an isochromosome. Isochromosomes are chromosomes that either have two P arms stuck together or two Q arms stuck together. This picture illustrates an isochromosome of X having two Q arms of X stuck together. Well, how could that happen? Remember in meiosis, when we have the sister chromatids joined together, ordinarily what happens when that cell divides is that those two chromatids split along a vertical line between them so that each daughter cell gets a complete copy of the chromosome. But every once in a while, for reasons we don't understand, that chromosome can split along a horizontal line so that one daughter cell will get the two P arms stuck together. The other daughter cell will get the two Q arms stuck together. Those are both isochromosomes.